so don't worry. Well, so we have started with the chapter of sexual reproduction in flowering plants. You can, Shweta. Yeah, you can. If you want, you can. That's fine. That's up to you. If you want to make a new notebook for it, it's your wish. Okay, because in total you have to write down the answers for 38 chapters altogether, including your 11th and 12th. So if you want to have as a separate book, you can. Checking doubts now? Yes, beta. Good. So start the work so that you get time to clear everything that is there in your mind about any of the chapters that we have completed. Okay, well, so we go ahead with the chapter called as sexual reproduction in flowering plants. And here, let me share the screen with you all. Okay, so in the last class we had seen till here, if I'm not wrong, I'll just show you the previous slide also. To complete all 7 chapters, but to submit only 4. Yes, bacha. Not only 7, whatever chapters we are going on completing now, you can start solving eventually. But now since there is a backlog, I'm giving you time to complete it. So that is the reason I told you that 15, you can complete your 11th standard ke 4 chapters and submit it. And then after, a week or 2, we can think about the next 3 chapters. And as in when we go on completing each and every chapter now, you all can start solving it side by side. No, not yet. Okay, so here now beta. So here we are completed till here, right? Microsporogenesis we had started and we had completed that, how the spores, microspores or pollen grain are going to be formed. So here we have seen simultaneous types, successive types, the diagrams we saw, and the 4 different arrangements. So tetrahedral would be one towards the back side. That's called as a tetrahedral. Isobilateral on the same axis. Decussate, 2 at the आगे पीछे और 2 उसके ऊपर, right? T shape very clearly seen 1, 2, 3, 4, and linear is in 1 row. We had completed up till here, right? Linear is in 1 row. So any confusion with whatever we have done till now with this chapter? किसी को कोई point नहीं समझा हो? All are okay with whatever we have done? Anther walls, epidermis, endothecium, middle layer, tapetum, everything okay? Well, so stomium, I'll tell you, don't worry. Wait. Where we had seen it. In between the cells of the endothecium, a few cells are going to be present without any thickenings. Okay, and the thick-walled cells collectively are going to be forming a stomium. So stomium is nothing but, as I had told you, the perforated dots of your checkbook, how it is. In the same way, there's going to be a scope for rupturing of the anther for liberating the pollen grains. So that particular line of dehiscence, right, is going to be called as your stomium. Got it? Inflorescence means arrangement of flowers on the floral axis. What is inflorescence? Arrangement of flowers on the floral axis. How are they arranged? So you might have seen certain flowers who are in bunches now. They are arranged in bunches. Some are single solitary flowers. Some are going to be one right, one left, one right, one left. You have seen such type of floral arrangement. Flowers की arrangement उस axis पे, उस arrangement को हम कहते हैं inflorescence. ठीक है? Got it? I think you might have seen those orange color ke flowers which are seen generally everywhere. They are generally seen like a bunch of flowers. या फिर if you have seen the flowers of mogra, mogra का देखा है आपने flowers? They are always in a bunch. So the arrangement is different in different flowers. So that arrangement is going to be called as inflorescence. Okay. Well, so stomium understood now, my dear? You okay with that word stomium now? Can we go ahead? Anyone having any other type of doubt? Please let me know. नहीं आगे जाए नहीं बॉक्स आ गया उसको पहले हटाते हैं ना डिस्टर्ब करता रहता है वो बॉक्स हमें उसको हटा दो चल फाइन so now we go ahead with the pollen grain structure. Basically, how does it look like? So pollen grain is also called as male gametophyte. Phyte, P-H-Y-T-E का मतलब होता है होल्डिंग, जो गैमीट को होल्ड करेगा, जो गैमीट को बनाएगा, उसको हम कहते हैं गैमेटोफाइट. सो स्पोरोफाइट, the one which is going to hold the spores. Gametophyte, the one which is going to hold the gametes. Okay. Do we have to learn examples? No. No need of examples. Only those examples which I'll tell you now. Only remember those. No need of other examples. Okay. So till now, whatever examples for this chapter have been told to you is just for reference. Right? Okay. So microspore or pollen grain is the first cell of the male gametophyte, which is partially developed. Why do we call them as partially developed? Because still the male gametes have not been formed. Pollen grain is formed, but pollen grain के अंदर male gametes बनेंगे, वो अभी तक नहीं बने हैं, इसलिए हम उसको क्या कहते हैं? Partially formed gametophyte. Okay? Please understand these terms मत, नहीं तो तुम जब read करोगे ना टेक्स्ट बुक का तो बहुत difficult हो जाएगा समझना. So just try to understand. What did I say? Gametophyte means the one which makes and holds the gametes. So pollen grain के अंदर दो male gametes बनने वाले हैं. Therefore, the pollen grain will be called as what? Gametophyte. So the pollen grain will contain two male gametes. Fine. And therefore, what we call the pollen grain as partially developed, because till now I haven't told you na that how two male gametes are going to be formed. Have I told you? No. So once I tell you that how the two male gametes are going to be formed, then it will be a completely developed male gametophyte. Initially, it is partially developed male gametophyte. Yeah, partially developed. Correct. Size is 25 to 50 micrometer. It is unicellular and haploid. Always pollen grains are unicellular and haploid. Haploid means you already know they're going to have only one set of chromosomes because they are a product of meiosis. They are a product of meiosis. So meiosis you already know. What is it? Meiosis 1, meiosis 2 to give us 4 haploid gametes. So it's going to be a haploid structure. The shape of the pollen grain. First cell of male gametophyte. After the formation of the pollen grain, only then will the gametes form. So till the gametes are formed, it is going to be the first cell, a single cell containing the capacity to make male gametes. Containing the capacity to make male gametes. Correct? So it's the first cell. First cell, one single structure, one haploid cell. Okay? Got it now? Well, the shape varies from oval to polyhedral because of the presence of the sporopollenin. हमने already देखा था, right? The wall of the pollen grain is made up of 2 layers. The outer will be called as enzyme. The enzyme is made up of sporopollenin, which is derived from a carotenoid, one of the most resistant organic material known. carotenoids are nothing but they are pigments. What are carotenoids? They are pigments. So sporopollenin is derived from the pigments called as carotenoids. And it, you know, I told you, it's resistant to heat, resistant to acids, resistant to alkalis, resistant to temperature, low or high. It does not have any effect of any of the enzymes. So sporopollenin is one of the toughest, most resistant substance. Okay, organic substance means, what do you mean by the word organic? The substance containing carbon. So जहां जहां carbon present है उसको हम कहते हैं organic substance. Correct? Yes, yes, yes. Vedant, correct beta. Bilkul correct answer. So that is going to be your organic. It is thick and ornamented. Who is thick and ornamented? The enzyme. Thick and ornamented मतलब कभी कभी spiny है, कभी कभी smooth है, कभी ribbon shape है, अलग अलग type के outer layers होते हैं enzyme. ठीक है? At certain places, now the most important point, at certain places, enzyme remains unthickened. Means there is no presence of sporopollenin in that area. Okay, and these places are known as germ pores. What are they going to be called as? Germ pores. So what are germ pores? The part of the enzyme which is not having any sporopollenin deposition. That particular part will be called as germ pore. I'll show you what is a germ pore eventually. Don't worry. Okay, here it's not there. I'll draw it for you all and I'll show you. Just I'll show you how the germ pore looks like. Okay. Sure. Yeah. So here you can see the whiteboard, all of you. Now I'll show you the structure of the pollen group. Okay. ठीक है? Okay. अब देखो. Here I'm drawing something like this. ये देखो. ये है ना, मैं ऊपर ये कलर अलग ले लूं तो इजी हो जाएगा तुम्हें इंटाइन एंड एक्साइन. एक मिनट में ना कलर चेंज करती हूँ तो तुम्हें ना इंटाइन एक्साइन बहुत क्लियरली समझ में आ जाए. So I'm making, see, I'm purposely making it, you know, irregular and spiny. It could be, you know, anyways उसका शेप कैसे भी होता है एक्साइन का, कभी कभी स्मूथ भी होता है, just like this. Okay, anyways the shape is going to be fine. Now here they're going to have what? They're going to have one nucleus. I'll draw the nucleus also for you all. So they're going to have a haploid nucleus. Okay, now I'll tell you the labels. You can see the red one, you can see the blue one, right? Here you can see a gap. Can you see a gap? Here it's going to be your one. Okay, now let us label them. This is the enzyme, which is going to be made up of what, beta? Sporopollenin se bana hai. Okay, so this is going to be made up of sporopollenin, right? This one, yeah, correct. Yes, Jaya, correct answer, beta. Bilkul correct. The other one, the blue one, is going to be called as, intine, and the intine is going to be made up of, what is it going to be made up of? A substance called as pectin, and it's also going to have a little bit of cellulose. So it's called as pectocellulose. Okay, pectin plus cellulose. So, you know, collectively it can be called as what? lactocellulose, pectin plus cellulose. Then here you can see a gap in between, and this gap is going to be called as germ pore. How many germ pores can you see? कितने हैं इस डायग्राम में? आपको कितने germ pores दिखाई दे रहे हैं? Yes, correct. So 3 germ pores can be seen. Okay. Well, and here you can see a structure which is nothing but the nucleus. Did you understand that how the pollen grain actually looks like under the microscope? हाँ, exine, बीच में gap है. Intine is entire. Can you see the blue circle is entire? उधर मैंने gaps नहीं बनाए हैं. So the intine is entire. Exine, बीच में gaps है. The gaps is going to be called as germ pore. Okay. Now something more. Nucleus has nucleolus. Everything. We have everything. Okay. In monocots, generally you will have one single germ pore. इसलिए उसको हम कहते हैं monocolpate. What do you mean by the word monocolpate? Having single one germ pore. Monocolpate. ठीक है. On the other hand, the dicots would have generally 3 germ pores, and therefore it's going to be called as tricolpate. Okay, fine. So yeah, gap is compulsory. Yes, Vedant. Without the gap, आगे की कहानी तो अब समझेगे ना, तब समझेगा कि why it is important. Yeah, gap has to be there. Okay. Intine function same like cell wall? No. There is something different and very important that the intine is going to do for us. Okay. What is the? I'll tell you. I'll tell you the function of germ pore. I'll tell you. Germ pores के बगैर आगे का कुछ कहानी खत्म ही नहीं होगा, मतलब चालू ही नहीं होगा. खत्म की क्या बात कर रही हूँ? It won't even start if the germ pores aren't there. Okay. I'll tell you the function. Just remember, fertilization gap? No. Significance of different number of germ pores. Significance is nothing but it's been observed that generally monocot seeds now only one opening is more than sufficient. But for dicot seeds, dicot pollen, it's going to be always more than 2. So generally 3 would be present. So it has been observed in general. Fine. How is germ pore formed? By non-deposition of sporopollenin. There will be some area which is not going to be having sporopollenin present. So if there is no deposition of sporopollen in a particular area, it will lead to the formation of germ pore. Right? So it's a gap between the exine. So exine is interrupted by germ pores. Is also, like some books call it like that way, that the exine is interrupted by germ pores. ठीक है? ये please लिख लोगे बेटा, because I have not mentioned these things in your PPT. I just want you all to write down this. Kindly draw the diagram. See, exine could be any shape. Noted? Very good, Divya. Very good. So the exine could be of any shape, okay? Because it's the what we say, decoration, Yubish bodies. You remember, na? Yeah. So that could be because of the shapes could be anything. Done? Okay. Those who have done, very good. I'll wait for 2 minutes for others to copy down this very quickly. See, my dears, it's always better that if you have got color pencils with you now, use color pencils to draw the diagram in your notes or your books, not in the examination, so that, it becomes very easy because of the distinguishing colors that what are you talking about actually. So generally, I tell all my students to buy that small, chota sa color pencil wala, ek chota box. So ना diagrams जो बनाते हो ना easily detect हो जाएंगे. See now that red and blue ना, तुरंत पता चलेगा. Red pens, okay. See, for your reference, in your board examinations too, you have to make all the diagrams with your pencil only, normal pencil. But for your reference, it is always better that you draw artistic diagrams. Artistic matlab colorful diagrams. I don't want it to be like, typical artistic. Pen bhi chalega, yeah, pen or pencil or whatever. Whatever is better for you all. Whatever you're comfortable with. Anything will. Well, I hope you all know Friday is going to be a holiday because of Ganesh festival. Huh? It takes sustain, my dears. When you will, like again and again go through it now, it will be easily coming in your mind. Don't worry. And I'll take revisions for you all. Yeah, Sanika, correct. Dicots, you want me to explain again? Yes. Dicots will have, the diagram which I have drawn is of the dicot pollen. Because it's having 3 germ pores, 1, 2, and 3. So whenever there are going to be 3 gaps in the enzyme, it's going to be generally the pollen grain of dicot plants. And if there is a single germ pore opening, it's going to be generally of the monocot plants. Monocot means having 1 cotyledon. Dicot means having 2 cotyledons. So chana है ना? Bean है. खाते हो कि नहीं ये सब कुछ? It's good for health. हाँ, छोले, पूरी नहीं रे, सॉरी, वो छोले के बाद ना पूरी आ जाता है माउथ में. वो छोले, पूरी नहीं रे बाबा. But puri is made up of wheat. Wheat is a monocot. So, yeah, lobia, that is in Marathi we call it chauri. You might have seen that with that black color का होता है उसमें. चौली होती है, चना होता है, छोले, राजमा, फिर your singdana, groundnut, they all are dicots. The ones which have got, when you break, you can have 2 equal pieces. That's going to be, pea also, yes, how can I forget pea? Moong, yes, moong is also a dicot. Matki is also a dicot. All of these, yeah, all of these are dicots only. So, cotyledons means the the seed which is going to be formed. If it is divided equally into 2 parts, the 2 parts each will be called as one cotyledon. So if you have seen singdana, normal सिंगदाना देखा है? हाँ, तो सिंगदाने को जब तुम खाते हो ना, तो अगर आप उसको ओपन करोगे तो आपको 2 पार्ट्स मिलेंगे उसके, है ना? So each part will be called as 1 cotyledon. Nuts, yeah, even nuts, some of the nuts, almonds, right? You get 2 cotyledons. Yeah, it breaks into 2, yes. When we roast the singdana, it breaks into 2. You know why singdana is coming in my mind again and again? Because Shravan जो होता है ना हमारा, उसमें maximum fasting होता है. Almost every second day I'm having fasting. तो fasting के दिन ना मैं बहुत ज़्यादा सिंगदाना खाती हूँ. तो जब भी मैं खाती हूँ ना, तो दिमाग में डाइकॉट का एग्जाम्पल आ जाता है. What to do? बायो टीचर हूँ ना, so right? तो कल अगेन टुमारो ऑल्सो इज अ फास्ट. सो अब भी मैंने वही बताया, लाइक आई टोल्ड कि वो सिंगदाना थोड़ा सेक के लेना पड़ेगा आज शाम को. कल वापस सिंगदाना खाना है ना? कॉन्जिक्यूटिव फास्टिंग ना. अरे यार नो यार, स्मॉल पीस ना, या दैट स्मॉल पीस इज गोइंग टू बी द ग्रोइंग टिप. सॉल्टेड नहीं, आई एक नॉर्मल वाला. सॉल्टेड नहीं अच्छा लगता है. लाइन अप द एंटायर मंथ. आई नो पूरा श्रावण है ना, फास्टिंग में ही चला जाता है. You ate salted one? So now next time whenever you eat singdana na, तो दिमाग में रखो, this is an example of dicot. Yeah, groundnut contains cholesterol, so you can soak it, and you can actually cook it. After cooking na, the cholesterol will go off. It will get enriched with vitamin B12. So generally ना सिंगदाना जो है ना, it should not be roasted and eaten, और कच्चा नहीं खाना चाहिए. उसको सीधी सीधी करा दो उसकी, तो उसका विटामिन B12 कंटेंट बहुत बढ़ जाएगा और कोलेस्ट्रॉल पूरा निकल जाएगा. देन इट इज वेरी गुड फॉर हेल्थ. आई ऑलवेज रीड लाइक दैट वे ओनली. Boiled, yeah, boiled one. You can take that entire groundnut ka pura shell and you can, just cook it. And that also tastes very nicely. You never knew that? Yeah, that's the key to eat, singdana with enriched properties of vitamin B12. So whenever you cook it now, the cholesterol will go. You will never have problem of cholesterol if you have boiled the singdana. Tell again, nothing. I just put everything in a cooker और उसको 2-3 सीटी लगाकर खा लेती हूँ. नमक डालती हूँ थोड़ा सा अगर बॉईल करूंगी तो. बॉईल मतलब कुकर में डालना है, 2-3 सीटी करोगे तो हो जाता है. But it gets enriched with vitamin B12. It's very good. Then it becomes healthy food. देखो. So bio सीखते सीखते gives it like that. Na, yeah. Generally, like, see, we mothers now, we know what is good for our kids. Dialogue na. Go out for Ganpati. No, in my busy schedule, it becomes very difficult. Even if I go out, maybe in the evening. Morning तो not possible only. तुम्हारे घर पे किसी के अगर आते हैं गणेश जी तो फिर तुम लोग अभी तैयारी में लग जाओगे, है ना? आजकल 2-3 दिन में विलेज जा रहे हो, गुड, एंजॉय. बट फ्राइडे लेक्चर ओनली. आई एम श्योर कि फ्राइडे का लेक्चर नहीं है. आई डोंट नो अबाउट अदर डेज. यू गेट कंफर्म फ्रॉम मास्टर क्लास एडमिन पीपल. Okay, so I hope you all have drawn the diagram by now, and you're not going to go out anywhere. Only Friday. I don't know. I don't know what other days. I know only because my lecture is on Friday, and I have been told that this Friday being Ganesh festival, there is no lecture karke. So I know only for Friday. I don't know about any other days. That you get it checked. Okay. So I hope you all have understood what is this diagram altogether. हाँ, परफेक्टली समझ में आ गया, राइट? अब है ना, मैं इसको हटाऊंगी नहीं क्योंकि उसके आगे आई एम गोइंग टू टेल यू समथिंग वेरी इंपॉर्टेंट, बट आफ्टर आई कम टू दैट पर्टिकुलर पॉइंट. सो लेट मी कम टू दैट पॉइंट अगेन. वेयर एम आई? Okay, so now you know what I'm talking about. So you know now what are the germ pores. Now you could recollect this. Huh? So at certain places, the enzyme remains unthickened or missing, and these places are going to be known as germ pores. Sporopollen is resistant to physical and biological. After the usual features, this is there. Sporopollen is resistant to physical and biological decomposition, alkali, acids. So pollen wall can be preserved for long periods in fossil deposits. So, like we have got many fossil deposits wherein the pollen grains are still intact. The enzyme exhibits a fascinating array of different designs and patterns. So here you can see at the base of this slide, the diagrams which are showing these, look at the wall, how it is. It is spiny over here. Here it's going to be, the way we made it, not entire. Here it's very, very smooth. Can you see? So these are the different designs and patterns you can see for the pollen grain ka exam. The inner intine is thin, delicate, and is made up of cellulose and pectin or pectose. So that is the same. Pectin and pectose is the same. So we call it as what? Pectocellulose, which I have already mentioned earlier in the diagram. In insect pollinated flowers, the enzyme of the pollen grain is covered with a yellowish, viscous, and sticky substance called as pollen kit. My dears, kindly remember this word. What is pollen kit? Now, what happens because of this pollen kit? It's a sticky glue-like thing that is going to be, made up on the pollen. Okay, so viscous means sticky. As like हम लोग चिक्कट बोलते हैं ना, एकदम ऐसा चिपकने वाला, चिपचिपा, उसको कहते हैं sticky. And viscous is thick, not free flowing. Just like how I can tell you viscous. Uh, you have seen honey. Yeah, honey is also viscous, correct. Or you might have seen, you know, the rubber being formed, how it's going to be pouring like that way. Okay. Yeah, wait, I'll show you that. I'll show you the earlier slide. Just let me complete this. So it's going to be viscous and sticky. What a yellowish color fluid, which is going to be called as pollen kit. What is the reason for the sticky fluid? See, this pollen kit perhaps is the protective envelope which also sticks to the body of the insects and thus helps in pollination. So when the insect is going to be coming on one of the flowers, okay, it's going to take the nectar. Now from the body parts of the insect, these pollen grains can get, you know, they can stick to the body parts of the insects. Because of what? Because of the sticky pollen kits. Okay, so it's going to help in what? Moving from one flower to another flower. When it's going to move, it's going to deposit those pollens over there. So just like helping in pollination, right? This is perhaps, okay, it is chiefly made up of lipids and carotenoids. So I just now told you, carotenoids are pigments. So it's going to have pigments and lipids. Who? Pollen kit. The development of the male gametophyte from pollen grain is called microgametogenesis. So microsporogenesis was formation of pollen grain, and microgametogenesis is formation of gametes inside the pollen grain. Got it? Understood? Yeah, you want me to show the previous slide for a minute? Okay. See this. Fine. So understood about the pollen grain or the male gametophyte, as we say. Any difficulty with the first slide or the second slide? Any point not understood of this particular thing? Yeah, beta. Welcome, my dear. Pura samjha? Nice. Okay, now we talk about the pre-pollination development. So before the transfer of the pollen onto the stigma takes place, what are the events or what are the different types of developments that are going to be taking place? Let us talk about them. Okay, see, a very important point now. This is a very important point, beta. Microspores, that is the pollen grains, start germinating in situ. Means there and there. Udar ke udar. That's called as in situ. That is while enclosed inside the microsporangium or the pollen sac. So sometimes what will happen? The germination of the pollen grain will start on the anther itself. That is called as in situ. We have already, I've told you already what is in situ and ex situ. Such type of development is going to be called as precocious. Precautious means in situ, उधर के उधर डेवलपमेंट स्टार्ट हो जाती है पोलन ग्रेन के अंदर क्या क्या होने वाला है उसकी. पोलन ग्रेन तो बन चुका है, it's already made. Now it has to move further to make two male gametes. So the process has begun for the male gamete formation when the pollen is still inside the anther. This type of development is going to be called as precautious. Anyone having difficulty with the first point? Got it very clearly? All of you, I'll repeat one more time. Precautious development means when the pollen grain is still inside the anther, the male gamete formation is initiated. This type of development is called as precautious. Microspores may be best defined as partially developed male gametophytes. I already told you what is the meaning of it. Producing male here, yeah. In case of precautious development, yes, the initiation of male gametogenesis has started, right? Microspore nucleus divides mitotically to form a smaller generative cell lying next to the spore wall and much larger vegetative cell, or it's also called as tube cell. So in the adjoining diagram, you can very well see बेटा, ये जो diagram है, देखो ये मैंने अलग से क्यों बनाया था, पोलन ग्रेन का डायग्राम है ना, बिकॉज यहाँ पर है ना जर्म पोर्स कहीं पर दिखाई नहीं दे रहे, यू कांट सी द जर्म पोर्स, राइट? So to make you all understand that what exactly are germ pores, I had drawn that particular diagram. Of course, they have made this nucleus inside. You can see it very clearly. Now, what are they saying? See, here, this is going to be the presence of vacuoles. So when the pollen grains will, become fully grown, then there will be formation of vacuoles also. Vacuoles are nothing. They are going to be spaces which are going to contain cell sap, or they could be having just air present in it. So it could be anything. Look at this. Now this is the first mitotic division, which is giving rise to one small and one large cell. Do you remember, recollect something called as, in females, there is something called as the secondary oocyte and the polar bodies? Very good. Yes, we have already written polar bodies. So जैसे human females में होता है ना, one large ovum or the secondary oocyte and one small छोटू polar body. Something like that you can see over here. One large vegetative cell, one छोटू generative cell. But यहाँ पर है ना, it's exactly opposite बेटा. The छोटू generative cell is ultimately going to make the male gametes. In our case, the larger secondary oocyte was involved in zygote formation. Here, the small chotu generative cell is going to be involved in making the male gametes. Okay, so here in this diagram, you can see very well. This is the large cell, which is the vegetative cell. This is a small cell, which is the generative cell. So there is unequal division over here in case of flowering plants when they make the male gametes. So pollen grains formed by meiosis. Pollen grain के अंदर अब दो मेल gametes बनाने के लिए mitosis. ठीक है? So this is the first mitotic division giving rise to two cells, one large, one small. Large called as the vegetative cell or the tube cell, small called as the generative cell. ठीक है? Vegetative cell contains large amount of food reserves and large irregularly shaped nucleus. Okay, the generative cell is small and it appears to actually float in the cytoplasm of the vegetative cell. It appears. Okay, it is spindle shaped and with dense cytoplasm and nucleus. Spindle समझते हो? You know what is a spindle? Spindle shape कैसे होता है, किसी को पता है? Is generative cell more nucleus as compared to the vegetative cell? No. Okay. Pointed at the ends. Yes. Spindle is going to be, like tapering at both the ends and bulged at the center. That's called as spindle. I'll tell you exactly. If you have ever been to a tailor shop, कभी गए हो? टेलर के शॉप में कभी गए हो? हाँ, तो उसके पास एक मशीन होती है, पैर हिला हिला कर है ना, वो मशीन को चलाता है. स्टिचिंग मशीन होती है, राइट? अब स्टिचिंग मशीन के ऊपर है ना, दे पुट द थ्रेड. यू हैव सीन? दे पुट द थ्रेड. तो थ्रेड कैसे होता है? सेंटर में पूरा थ्रेड है, ऊपर वो कार्डबोर्ड के जैसा एक सर्कल है, नीचे भी एक कार्डबोर्ड के जैसा, हाँ है ना, ऐसे दो ये होते हैं. So that is the spindle. Actually, that is called as a spindle. Tapering at the ends and bulging at the center. That's called as spindle shape. Okay. So even this, the apna generative cell na, it appears like a spindle shaped structure. Okay. If you look at your eye, how is it? Even convex lens. Correct. Understood now what is spindle? Exactly. It is spindle shaped with dense cytoplasm and nucleus. The pollen grains are shed. Now look at this. This is a two-celled structure of the pollen grain now. So pollen grain internally is going to have two cells, vegetative and generative, right? Now see, the pollen grain are shed from the anther at this bi-celled stage in over 60% of the angiosperms. In over 60% of the angiosperms. In the remaining species, now please understand, 60% of the angiosperms means there are 60% of the, types of angiospermic plants, flowering plants, in which pollen grain will be released, liberated, thrown out from the anther in this particular condition. Which condition? Two cells. Which are the two cells? Vegetative and generative, right? But what happens in rest of the 40 days? तो अभी तक मेल गैमीट तो बना नहीं है. Can you see anywhere? मेल गैमीट बना है क्या 60% of the angiosperms में? नहीं, दो cells है, vegetative and generative. Where is the male gamete? Not formed till now. Okay, but look at the rest 40% of the angiosperms. Just see, in remaining 40% species, generative cell divides mitotically one more time to give rise to two male gametes. Okay, okay, and when? Before the pollen grains are shed off, before the pollen grains are released, before the pollen grains are liberated out of the anther, उसके पहले ही 40% of the angiospermic species में क्या होता है बेटा? The generative cell will undergo one more mitotic division. So now the pollen grain will be having three cells. Which are the three cells? Two male gametes and one more vegetative cell. Correct? So look at this diagram over here, you will come to know. See, here there is mitotic spindle which is going to be taking place and it will give rise to two male gametes in 40%. But in 60%, at this stage only, the cell is going to get, the pollen is going to get shed off. Did you all understand? Two-celled and three-celled structure. Any confusion in this? What is two-celled and what is three-celled? Two-celled, vegetative cell plus generative cell. Three-celled, generative cell undergoes one more mitotic division to form two male gametes. Vegetative cell intact है और generative cell सिर्फ दो parts में divide हो गए. ठीक है? I told you that only now, right? So how are the three cells? Two male gametes and one vegetative cell. So it becomes three cells. So one male gamete is one cell, another male gamete is the second cell, and vegetative cell is the third cell. So now in this way we have got three-celled structure. So in 40% of the angiosperms now, all the three cells will be formed in situ. Okay. While in 60%, what is happening? See, it's only two cells. That means in 60% of the male gametes, in 60% of the angiosperms, male gametes बनेंगे ही नहीं क्या? What do you all think? बताओ. क्या लग रहा है तुम्हें? Will it like not make any male gametes? बनने चाहिए ना? क्या लग रहा है? चलो तुम लोग बोलो. तुम्हें क्या लग रहा है? बनने चाहिए कि नहीं बनने चाहिए? कि जेनरेटिव सेल ही फ्यूज करेगा एग के साथ. बनेंगे? They are going to be formed. Never ever it has happened that the male gametes have not been formed from the generative cell. ऐसा नहीं होता बेटा. Every time, let it be in situ or let it be ex situ. Now the word comes. Ex situ means not in the anther, somewhere else. Right? What is that somewhere else? I'll tell you later. Ideally fused, vegetative cell should have ideally fused so that the zygote can get nutrition. No, this is male gamete. Male gamete is never concerned with nutrition of the zygote. It's always the female gamete. Females. हाँ. घर पे खाना कौन देता है तुम्हें भाई? हाँ. कभी कभार पापा बनाएंगे तो बात अलग होती है. हाँ, कभी मॉर्निंग वाली चाय पापा बनाएंगे तो मम्मी बहुत खुश हो जाती है या नहीं? होता है ऐसा? बाय चांस बहुत बार? देन योर मम्मा इज लकी. You make for everyone. That's nice. And I'll come to your house to taste the tea of, made by you. That's great. Yeah, you make it in the evening. Chalo, then I'll come to your house in the evening. साल हाँ बहुत सालों में एक बार ना हाँ बेटा यही होता है so feeding the you know family or feeding the zygote it's concerned with the you know female side of the family generally. I don't drink tea. I'm sorry, but I don't drink tea at all. Yeah. Tree matlab I was actually, like there are people who are they cannot survive without tea. Have you all heard about such people? They drink tea anytime and every time. है ना? Addiction. Yeah, very word, perfect word. Yes, Sadika, it is addiction. You know, I was a typical addicted female of tea. In the night also at 2:30 hours to get up and has to make tea and have it. But and I feel I should have. And what used to happen, like, whenever I used to take lectures now, so in PACE, in Akash and all, now, whenever we had lectures now, so continuously I used to have, I mean, the peon, the office boy used to always carry tea. And within the lecture also, they used to knock and they used to, keep that chota cutting wala tea on my table. And I was so used to have, like tea every after half an hour. Every after half an hour I used to have tea. मतलब कम से कम मैं है ना पूरे दिन में है ना 20 to 25 cuttings चाय पीती थी. Imagine 20-25 cups of tea. I was like totally bad about it. I couldn't, like control myself of not having tea. And then like one fine day, acidity and all, I was okay with it. I love tea, so it's okay. So I was, like I couldn't sustain without tea. And typically, like that water भी नहीं पीती हो ना उतना. हाँ मैं उतना चाय पीती थी सच में कसम से. नो exaggeration. So बचपन से I was like a typical है ना. I did not have any problem with acidity or something. I had the capacity to digest it. But then, like one fine day, I just thought to myself that how much is my dependency on something? That's not good. You should not have so much dependency on, anything basically. So I thought to myself that enough of it. From tomorrow onwards, I'll quit tea. I took it on my own. No one forced me. No one told me. And I just took it on my, own will and own wish that I'm not going to have tea anymore. And from that day, I stopped having tea completely. Now it's past 5 years or so, 5 or 6 years that I have not had tea at all. I have stopped tea completely. Yeah, some people need it once in like, but I decided that no, I'm not going to have it. I'm not going to have it. Let me see my willpower. I challenged my own self. And when I challenge my own self, that's a habit. That's a habit. And it's a green tea, black tea, nothing. Headache, it's all in the mind. It's all in the mind. If you decide by your head and your heart that that much of willpower should be there in all of us. Otherwise, what is the difference between being a human and being an animal? Right? So always decide, but always challenge yourselves only. Don't challenge others. Others kya karne ka hai? Challenge yourselves. I'm going to do it. I'm going to do it. I'm not going to do it. I'm not going to do it. Everything is in the mind. If whatever you think now, you will accordingly behave. Yes, self-consciousness. Perfect. So that is my habit. And that's a very, I may not say it's a bad habit. Sometimes it's a good habit, but sometimes I become very rebellious against myself in these type of cases. Heart and mind, yeah, war goes on. Let the right thing win, Yeah, self-control is very important. That's good. That's really good. So, soft drinks as well. When I was in the field of teaching. See, because being in this medicine field na, zoology was my forte. Zoology में कुछ भी पढ़ाना is like simple. I mean, what is difficult in human anatomy? It's like, it was my cup of tea. It was like simple. But when I started with teaching now, then I said that it's not like that way that I should only be a teacher of zoology. What is the use of it? I should be a teacher of biology. So 20 years before only I challenged myself that I'll become a master of botany as well. And I fought with myself. I took whatever books were required. I upgraded my knowledge from that time. And initially I struggled a lot with botany because such a big gap from 11th and 12th. Then after 5-7 years, when I'm again touching botany, it was like, oh my God. But then, I took it to my heart that I would be better known by, teaching the entire bio rather than teaching only zoology or only botany. And that is what is there in front of you right now. So right now I'm teaching you botany only, and I hope you all are understanding it equally. Hi, Anna. Are you all understanding? Yeah, I know. So that is the way it should be, I know, my dear. I know, because when even like I was in your place, when I was in 11th standard, 12th standard, I used to always wonder that why do I need botany to get into MBBS? What is the use of that botany, yaar? I mean, क्यों पढ़ना है उसको? But then, yeah, exactly. I know this. This is a common question. DR first. Okay. So, this is a common question that comes in everyone's mind, that why do we need to study botany? Hai na? But you know what, my dears? I'll tell you a very small, important point. That is, you all might be knowing how to ride a bicycle. है ना? सबको cycling आती है. I'm sure. सबको आती है ना cycling? है ना? हाँ. But एकदम से cycling आ गई क्या तुम्हें? You're a master in cycling. Very nice. Directly आ गई. मतलब मम्मी पापा ने साइकिल पे बिठा दिया और आप एकदम हाथ छोड़ के चलाने लगे फर्स्ट डे फर्स्ट शो. नो ना? ट्रेनिंग. एग्जैक्टली. 50 बार गिरेंगे तो ही सीखेंगे ना भाई मेरे. राइट? You had your normal 2 tires of your bicycle and there was additional support from the sides. Correct? Side supports होते हैं ना? And plus your mama or your daddy or your elder brother, elder sister, someone was holding you from behind so that my child does not fall. हाँ या ना? पीछे से पकड़ते हैं ना? और बाद में क्या करते हैं? अगर मम्मा या पापा आपको अगर पीछे से पकड़े हुए हैं, आपको लग रहा है पापा ने पीछे से पकड़ा, साइड सपोर्ट्स निकाल दिए, अभी आपके पेरेंट्स आपको होल्ड कर रहे हैं. हाँ, whatever, that's what I said, brother, mother, whoever it is, father, whoever it is, what do? Yes, all of a sudden you will realize, oh my God, no one is holding me and I can ride on my own. है या नहीं? वो वाला moment सबके लाइफ में आया है. That yes, now I can ride on my own. No one is holding me from behind. You get scared. Hurry, please hold me. I'll fall. You are not going to fall. That much of confidence the teacher already knows. Teacher, यहाँ पर the one who's going to teach you cycling. है ना? Yes, exactly. So in the same way, beta, whenever botany is going to be there, botany is like a side support. Botany is like your brother, mother, father who is holding you tightly, and one fine day they let you out, go on your own, the way that you want to cross, the way where you are heading for. You know why? Because your brain requires training, your brain requires nurturing, your brain requires practice. Okay, so unless and until you don't start, botany का learning, आगे जाके है ना, MBBS, BDS, फिजियोथेरेपी, जिसके लिए मगिंग अप लगता है, जिसके लिए टर्म्स दिमाग में रखने पड़ते हैं, जिसके लिए बार बार एक ही चीज पढ़नी पड़ती है, वो नहीं हो पाएगा बेटा. It's standard only that the training has begun for a career which you're going to make a name in after 7 years. So the training has begun from your 11th standard by teaching you all and making you all learn botany, my dear. Right? So botany is not going to be helpful as a knowledge probably in your MBBS or BDS or whatever you're going to go ahead with. But botany is definitely going to train your brain to start remembering the concepts, the names, the words, the terms which you have never heard of. So when you will actually enter into your destined colleges now, eventually after 2 years, 3 years, whatever, that time your brain is already trained. Now you want to, leave your hands free from the bicycle and ride on your own. The same way the botany is going to give you the confidence of learning, grasping, and taking in more information which you to make a career into. Yes, right? एग्जैक्टली सो प्लीज है ना, मैं पहले ही बता रही हूँ बेटा, लगेगा कि बॉटनी की क्या जरूरत है, लेकिन बॉटनी बहुत इंपॉर्टेंट है अगर आपको सक्सेसफुल करियर्स आगे बनाने हैं. आप कुछ भी बनो, यू गो फॉर BSc, you go for MSc, you go for PhD, you go for research, you go for pharmacy, you go for MBBS, you go for BDS, you go for physiotherapy. इतने बड़े बड़े बुक्स आएंगे पढ़ने के लिए. हाँ, अभी तक 10 तक क्या छोटे छोटे, right? So that is required. Why physics? Again, the same question which I had, but later on in your life, you will come to know that majority of our concepts are based on physics. Like when I teach you circulation chapter, you will come to know how the stethoscope works, how the blood pressure machine works, how the sonography machine is going to work. Eventually, we'll also use the sonography machine. Everything is required. So physics is also equally important. Yeah, some of the students now, they are more into making biomachineries for the or technological upgradations. You might have heard about Doppler machines. You might have heard about the MRI machines, which are actually medical. It's used for medical purposes, right? Magnetic resonance, है ना, MRI वाला, या फिर डॉपलर इफेक्ट वाले ये होते हैं, सोनोग्राफी के टेक्निक्स होते हैं. ऑल आर बेस्ड ऑन फिजिक्स. सो यू शुड हैव द नॉलेज ऑफ ऑपरेटिंग ऑल दीज थिंग्स. विदाउट द नॉलेज ऑफ फिजिक्स ना कुछ नहीं हो पाएगा, राइट? So physics is also equally important. Chemistry, though, you will only come to know why chemistry is important when I'll teach you photosynthesis and respiration chapter. You only will tell me, Miss, are you teaching chemistry to us now? Have you started teaching chemistry? When I teach you photosynthesis, you will ask me, Miss, are you teaching me physics? आएंगे, मैं वो भी आपको पढ़ाऊंगी, थोड़ा बहुत फिजिक्स भी पढ़ाऊंगी, थोड़ा बहुत केमिस्ट्री भी पढ़ाऊंगी, सब पढ़ाऊंगी. MRI checkup, yeah, that's what. So we should have the knowledge of everything, right? Yeah, see, everyone is going to, have these things, basic things common with them. All the subjects are interrelated. Yes, my dear Jaya, very correctly said. All the 3 subjects are interrelated and interconnected. You know what? During my time now, we had maths also compulsory. And I used to always wonder, why maths? चलो फिजिक्स ठीक है, केमिस्ट्री ठीक है, बायो तो चाहिए ही. Why maths? I used to literally cry out for that. But we had to do. We had no option for maths. Nowadays, we have got option for maths. Actually, but we had to do maths. There was no scope without maths. Anyways, but so that's good. That's good. If you love maths, your things will be, better understood with respect to physics. In fact, that's good. If you like maths, it's really good. Well. So I hope you all have understood this 2-celled and 3-celled structure very clearly. No problem with this. We'll move ahead. Anyone having any difficulty with 2-celled and 3-celled structure? Come on. Sure na? Aage jaye? Sure. Let's move ahead with the next point of this particular session. Yeah. Yeah, in the generative cell. Yes, beta. Only in the generative cell. Nothing will happen in the vegetative cell. Okay. The छोटू cell is the generative cell जिसमें mitosis होने वाला है. Now we come to the post-pollination development. Post-pollination का मतलब क्या है? Now the pollination has taken place. So the pollen grain has now come and it is now sitting, or it has been lodged on the stigma of the flower. We know gynoecium is going to be made up of stigma, style and ovary. तीन चीजें होती है ना, स्टिग्मा, स्टाइल, ओवरी, है ना. तो जो स्टिग्मा है, it is called as the landing platform for the pollen grain. Okay, so once the pollen grain is deposited on the stigma, we call it as what? Post pollination. So pollination has already occurred, and now what is going to happen? So let us see. The liberated pollen grains are transferred to the receptive surface of the carpel, that is nothing but the stigma, by the process called as pollination. On the stigma, the pollen grain absorbs water and swells within a few minutes. So what will the stigma do? Stigma will have a lot of moisture. It's going to have actually a sugar solution. It's going to have actually the sugar solution. And from the sugar solution, it's going to be absorbed by the pollen grain. ठीक है. अब पोलन ग्रेन जो है उसके अंदर वेजिटेटिव सेल है और जेनरेटिव सेल है, या फिर वेजिटेटिव सेल है और दो मेल गैमीट्स है, राइट. अब उसका वॉल्यूम बढ़ेगा कि नहीं? डोंट यू थिंक the volume will increase when it's going to start absorbing the sugar solution or the water, as they are saying? Don't you think it will bulge? Swell हो जाएगा कि नहीं? Correct? Swollen हो जाएगा कि नहीं? Exactly. So now what will happen, you know? Now what will happen? It releases the wall health recognition factors. And whether the pollen grain will germinate on the stigma or not. Means what will happen, I'll tell you. Suppose the stigma is of mango plant. The stigma is of mango plant. और इस स्टिग्मा पे राइस का पोलन ग्रेन आके गिरा. इसका पोलन ग्रेन आके गिर गया, राइस का पोलन ग्रेन आके गिर गया. अब पोलन ग्रेन ने क्या किया? राइस के पोलन ग्रेन ने क्या किया? एब्जॉर्प्शन करने को स्टार्ट कर लिया. बट द रिकॉग्निशन फैक्टर्स टू रिकॉग्नाइज is it of my type or is it of different type will stop the pollen grain from taking in that particular sugar solution, because it is not of the right type. Right type means if it was mango ka pollen, yes, it is of the right type. But ये pollen किसका आया है? Rice का pollen आया है. Be allowed to germinate on that stigma? No, not allowed. So who is going to decide that? The wall held recognition factors. How are they going to recognize that? कैसे recognition होता है? The carpel का structure and the female gametogenesis. आप भी नहीं समझेगा कुछ भी बोलूंगी तो. But there is someone which is present in the female, which is going to tell these recognition factors. Don't allow that stupid to drink that particular sugar solution. It is not of our type. Okay. Then they won't allow the sugar solution ka absorption if it is of different type. Huh? I got stuck. Did I? No, na. Acha, okay. Fine. Now it's okay. Maybe something with the net as usual. Audio was clear. Okay. Well, very good, Zui. I was expecting this question, that the wall factors are released by whom? I'll tell you that. वो होता है ना, एक सीरियल चलती रहती है बरसों से, एवरी डे हम उसको देखते हैं. क्या क्यों देखते हैं? आगे क्या होगा? आगे क्या होगा। तो एक पर्टिकुलर पॉइंट पे, जैसे कि ऐसे होता है ना, दे शो यू इन दैट हिंदी सीरियल्स और मराठी सीरियल्स और आई डोंट नो whatever serials are seen in your house, I don't know. So you know, someone opens the door and the person who has opened the door will be having a very big question mark, oh my god, how this person is over here, and then वो बंद कर देते हैं सीरियल को, and then they you have to wait for another 24 hours to watch that. Suspense maintain करके रखते हैं, वैसे ये सस्पेंस है। आज नहीं बताऊंगी मैं who is going to release the you know, recognition factors, वो suspense। I'll tell you definitely, but I am keeping as a suspense not for the reason of suspense, beta. It is being kept suspense because otherwise तुम्हें कुछ समझेगा नहीं what I'm going to tell you. No. I don't like TV. I don't have time only to watch TV. I only sit to watch matches, अपना cricket match. वो ही मेरा weak point है. News and cricket. Yeah, that is my weak point. Monday we won ना. Monday का देखा? We won that test. Yes, you're seriously good. I mean, I so like overwhelmed by the success on Monday. So nice. I don't like any sport, Are. I love this cricket. I like Olympics also. I like to watch football. I like to watch cricket. I like to watch all sports. How do they eliminate the different pollens? By recognizing it, if it's of the right type or not. So if it is of the right type, only then it is going to be allowed to drink that particular sugar solution. If it is not of the right type, the sugar solutions will not be allowed to get absorbed by the pollen grain. Okay, fine. Well, so these factors determine whether the pollen grain will germinate on the stigma or not. Germinate means whether it will be allowed to take in the solution or not. Okay. Subsequent to mutual recognition. So sometimes what happens is the pollen grain itself realizes that, oh, I have come on the wrong place. I should not germinate over here. So it's a mutual recognition. Both recognize each other in a mutual way. Mutual means together. Okay. So in this case, after they both recognize each other, yes, this is the right type. Yes, this is the right type. Only then what will happen? The vegetative cell enlarges because अंदर ले लिया sugar solution, तो मोटा हो गया वो, है ना? So it will bulge. Now pressure को release करना चाहिए नहीं. It can't afford to burst open. If it bursts, the male gametes will be lost on the stigma only. How will they reach the egg? They have to reach the egg. So they cannot afford to burst. So what will happen? The pressure of the sugar solution connection issue. Some connection issue. Now it's fine. So where was I? Thank you. So the pressure of the sugar solution is so high. The pressure of the sugar solution is so high that it's going to be released, right? And from where it's going to be released? So it's going to be released from one of the germ pores. So you know what are germ pores now, right? So of the 3 germ pores, one of them will be used for releasing the pressure. And the more it will drink the solution, more pressure will be built up. More is the pressure, more will be the elongation of the intine. More will be the elongation of the intine, longer would be the pollen tube. Understood what is the pollen tube? Now you understood very clearly what is the pollen tube. So what is pollen tube? Nothing, it's the extension of the intine. Correct? It's the extension of the intine. And from where the intine has extended? From the germ pores. So in the beginning, you had asked me, some of you had asked me, what is the function of germ pores? Now you understood the function of germ pores. Without the germ pores, pollen tube will not be formed only. And if no pollen tube is formed, how will the two male gametes reach the egg? इसलिए मैंने बोला था कि without pollen tube, मतलब without the germ pores, तो ये कहानी खत्म क्या चालू भी नहीं हो सकती है. आप समझा बेटा? हाँ. So see, subsequent to mutual recognition, where is my pointer? I can't see it. Subsequent to mutual recognition, the vegetative cell or the tube cell enlarges and comes out through one of the apertures, that is the germ pores, in the form of pollen tube. The wall of the pollen tube is nothing, but it is the extension of the intine. Correct? The tube secretes exogenous pectinases. You already know that the intine is made up of pectin. Huh? So the tube secretes exogenous, means uske bahar, tube ke bahar. Huh? Pectinases and other hydrolytic enzymes to create a passage for its entry. Kidhar enter karega wo beta? So from the stigma, that pollen tube will start piercing, entering the style. है ना? तो style के tissue को उसको dissolve करना है. So the pollen tube will start making pectinase, which will dissolve the pectin present in the style. Got it? And वो अपना खुद का रास्ता बनाता जाएगा. जैसे you might have seen the earthworm makes holes in the soil, na? Right? By secreting certain enzymes out of its body, it is going to make holes in the soil. In the same way, this pollen tube will start secreting enzyme called as pectinase, which will destroy pectin of the style. And through that pore, now the pollen tube is going down and it's trying to reach the egg. Okay? Got this? What's your doubt, beta? So the pollen grain relieves the wall held recognition factors. No, pollen grains won't. Exogenous means outside. Exo means outside. Genus is generation. So the enzyme called as pectinase will be made to move outside the pollen tube, causing the destruction of the tissue of the style. Got it? Now understood very clearly? बाहर की तरफ पेक्टिनेज बनता है, पोलन ट्यूब के बाहर की तरफ पेक्टिनेज बनेगा जो स्टाइल के टिश्यू को डिसॉल्व कर देगा और वो पोलन ट्यूब उसके अंदर 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 डीप 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 जा पाएगी. So to make a passage, to make a pathway for the pollen tube, it is very difficult because style is there in between. Style is made up of, pectin tissue containing pectin. So it is difficult to pierce through the style. So what is the easier way? जिसका स्टाइल बना है, जिस टिश्यू का स्टाइल बना है, उस टिश्यू के ही केमिकल को डिसॉल्व कर दो. करेक्ट? तो एक पैसेज बना, एक हॉलो ट्यूब बनी और उस हॉलो ट्यूब में से अब पोलन ट्यूब पास हो रही है. Just like how we have got these tunnels. You know what are tunnels? Huh? There's a huge mountain. What do? They make a gap at the center of the mountain so that you can pass from this area to that area via the tunnel. Same like that way. Huh? How did they find out all these? Nah? See, it's so interesting. Nah? It was found out by Navas Chin. It will come later on. It's come later on. There was a person called as Navasjan who has found out all these things. Great now. Yeah. So you understood what is happening now? Asian. Okay. So the wall of pollen tube is going to be the extension of enzymes. The tube secretes exogenous pectinase and other hydrolytic enzymes to create a passage for its entry. एक tunnel के जैसा. It absorbs nourishment from the transmitting tissue of the style. Transmitting means it is moving across the style by dissolving the pectin, right? So it's going to derive the nourishment from the style. Who? The pollen tube. Did you all understand what has happened till now in post-pollination development? Pollen grains came, they were dusted onto the stigma of the flower. If it was of the wrong type, it was not allowed to absorb the sugar solution. If it is of the right type, it is allowed to absorb the sugar solution. When the sugar solution was absorbed by the pollen grain, it started swelling. There was tremendous pressure developed inside the vegetative cell. So it extended through one of the germ pores. It came out via one of the germ pores as pollen tube. So pollen tube is nothing but it is the extension of the enzyme. Now this pollen tube had to cross the stigma and enter inside the style. But the style was having tissues which was not allowing the passage of this pollen tube. So the pollen tube started making enzyme called as pectinase exogenously, means outside the pollen tube, the pectinases were released. Now, because of the pectinase, the style tissue got dissolved, making a tunnel. Okay, and it became easy for the pollen tube to penetrate deep into the style through that tunnel. The nourishment that was required for the pollen tube to germinate, grow, extend was provided by the style tissue. Okay, this is what has happened in this particular slide. Any doubts, any difficulties in understanding post-pollination development? Anyone who did not understand this? Any point? Come on, go through it, go through it. Who is going to make those wall recognition factors is still a question mark, but वो थोड़ा suspense बनाए रखूंगी. Okay, got it, all of you? Sure? Okay. Pectin is a substance which is going to be present in the plant. Okay, and pectinase is an enzyme that will dissolve the pectin, that will degrade the pectin. So the style tissue is also going to have pectin in it. So the pollen tube will start making an enzyme that is going to dissolve the pectin of the style. Okay, clear? So just like you have got cellulose, to break down cellulose, you require cellulase enzyme. Similarly, there's another chemical called as pectin. To degrade pectin, you require pectinase enzyme. Okay, got it? Pectin is going to give rigidity. If you have seen the fruits, okay, if you have like you eat fruits, you might have had mangoes or banana, the pear, anything that you want to consume. Na, kabhi hai na, if you overripe it, okay, if it gets over ripened है ना, so वहाँ का एक part जो होता है ना वो पचक जाता है, देखा है, soggy हो जाता है पूरा. क्यों soggy हो गया बेटा? Because वहाँ का pectin dissolve हो गया. On over ripening na, the pectin is going to get degraded, and as the pectin gets degraded, the strength, rigidity, integration of that particular structure is going to get damaged. So easily you just put your thumb over there na, it goes inside. That is because of the degradation of the pectin. Huh? Yeah, pectinase dissolves the pectin in the style. Yes, Vaishnavi, bilkul correct. Pectin, yeah. Pectin is going to keep it fresh, exactly. So भिंडी देखिए आपने, भिंडी की सब्जी खाते हो तो भिंडी फ्रेश है या नहीं है कैसे चेक करते हैं? कभी किया है? आज ही खाइए. How do you check if the bhindi is fresh or not when you go to that sabji mandi to get it? Have you all ever tried? Breaking the end, है ना? That tapering end को आप break करते हो. And if it breaks quickly, that means it is fresh. Means what do you mean by it is fresh? It is loaded with pectin. If it does not break ना, ripen हो चुकी है वो, उसका pectin degrade हो गया है, वो अभी पचक जाएगी थोड़ी ही देर में, right? So pectin is going to give freshness, correct? Fine ना? Understood up till here, beta? Yeah, crunchy. Yes, Palak, very correct. It gives that crunchiness. Very right. Very right. Huh? Okay. Anyone else having any difficulty with this post-pollination development? They do give it nowadays. Artificially, they give everything to have them, a longer shelf life. So to maintain their shelf life, they give even hormones, different hormones injections. We dete hai. You can't. All like sometimes there are so many hormones which you want to go ahead with. आगे के चैप्टर्स में है 5 हॉर्मोन्स हमें प्लांट्स के भी स्टडी करने हैं और ह्यूमन के तो करने ही है. So that time you will come to know that plant के hormones play how much vital role in the freshness of the fruits, vegetables and all. Well, I understood this post-pollination. Then see what happens. Gradually, the vegetative and the generative nuclei are carried by the pollen tube. The former lying at its tip. Means the vegetative nucleus is going to be at the forefront. उसके पीछे 2 male gametes. तो वो pollen tube जब हम देखते हैं ना under the microscope, you will see 3 nuclei. One nuclei will be of the vegetative cell or the tube cell, which is going to be the leading one. और उसके पीछे you will see 2 more nuclei, which are going to be of the male gametes. So in this way, you can see 3 in 1 row in that pollen tube. Okay. The generative cell divides to form 2 non-motile gametes in the 60% of the angiosperms, which were, I had asked you na, that इसका मतलब 60% of the angiosperms में generative cell divide आगे करेगा कि नहीं करेगा, male gametes आगे बनेंगे या नहीं बनेंगे, तो अब बनते हैं बेटा. So the second mitotic division जो होता है ना generative cell का in the 60% of the angiosperms is going to take place inside the or on the stigma. इसलिए it is called as XC2. In C2 was 40%. XC2 is going to be 60%. So the rest of the 60% of the pollen grains which are shed at 2-celled stage will undergo the second mitotic division in the generative cell only, but not in the anther, rather on the stigma. Got it? So ultimately, the pollen tube, let it be 2-celled or 3-celled, ultimately the pollen grain will carry 3 things. What are the 3 things? First, tube nucleus. Second and third, 2 male gametes. Got it? Clear? This sentence is clear? Wait, It's, yeah, that's true, Vedant. Agreed. Right? Okay. Now, the next is what? See, the tube nucleus has no important function and may disintegrate. Pollen tube में tube nucleus का जो nucleus है उसका कुछ specific काम नहीं है. Right? Pollen grains of many species cause several allergies, bronchial afflictions, leading to chronic respiratory disorders like asthma, bronchitis, etc. So sometimes, these pollen grains are allergic. Some people get allergy when they come in contact with the pollen grains, like Parthenium grass. Parthenium is a grass which is allergic. So what is allergy basically? People will start sneezing. They'll have running nose. They have redness of the nose and eyes. Water will be coming from their eyes also. किसी किसी को अलग अलग एलर्जीज होती है. हाँ, hay fever. Yes, yes, very correct. Hay fever. Some people have got weird allergies. है ना? So in here also ना, sometimes the pollen allergy is a very common thing in some people. Okay? Pollen grains are rich in nutrients. Pollen tablets, pollen syrups are used as food supplements. So many Western countries, they make pollen tablets, pollen capsules basically, and pollen syrup to give strength and stamina. So it's going to be, acting as a food supplement. It's very rich. Pollen grains are very rich in nutrition. Okay. Pollen consumption increases the performances of athletes and race horses. So it's going to be rich in nutrients. So it's, made to the horses, the race horses are fed on rich diet of pollen grains. So that, they get strength, vigor, stamina to run the race and, get the top positions. Even athletes consume these pollen tablets and pollen syrups. Okay. How? We saw now generative cell, tube cell, two male gametes. More is the number of nuclei present in one cell. More would be the nourishing capacity, which we have seen in tapetum also. Tapetum was also multinucleate and it could nourish the developing pollen grains. So more is the number of nuclei, more would be the nutrition. Okay. So here they have given the functions of pollen. Okay. Pollen grains should be viable when they fall on stigma. Viable means capacity to germinate. What do you mean by the word viable? They should have the capacity to germinate. So pollen grains should be viable when they fall on stigma, so that may carry fertilization. If they cannot germinate, how will the fertilization be ensured? Right? Viability of pollen grain depends upon existing temperature and humidity. Please remember these two factors, that if the pollen will remain viable or not will be decided by the humidity and the temperature when they are going to be liberated. In rice, wheat, jowar, bajra, maize, sugarcane, okay, all of them, the pollen grains lose their viability in a short time span. That is only 30 minutes. So within 30 minutes, if the pollen does not fall on the stigma, no use. No use. It's dead. It cannot germinate now. So the germination has to be ensured within 40 minutes of its dispersal. Okay. So the viability of the pollen in case of rice, wheat, etc. is only within 40 minutes they have to germinate on the stigma. बहुत कम time hai, I know, right? But yeah, that is the way it has been recorded. Pollen grains may remain viable for months in plants belonging to families like Rosaceae, Leguminosae, and Solanaceae. So in the Living World chapter, we know A, C, E, A, E is the suffix for families, right? We have already learned this, right? So Rosaceae, of course, as the name says, the rose family, right? Leguminosae, the legume family, all the legumes. What are the legumes? Chana, pea, tuvar, choli, moong, all are going to be belonging to Leguminosae. And Solanaceae is the potato family. You know what is Solanaceae? Yes. Solanum, Petunia, and Datura. Yeah, it's called as the potato family. So the families of, the pollen grains of these three families is going to be having a viability for months, while rice, wheat, etc. is going to be only 30 minutes. Okay. In many plants, pollen grains can be stored in liquid nitrogen at -196 degrees Celsius for years all together and can be used as pollen banks like seed banks. So nowadays, you have got this technology of storing pollen grains for years together. And whenever required, those pollen grains can be taken and used for artificial germination. Okay. Such stored pollen grains can be used in plant breeding programs. So nowadays we have got these pollen banks also, just as we have got seed banks, blood bank, like that way we have got pollen bank as well now. Got it? यहाँ तक कहानी समझ में आ रही है? ये simple है, but still. Okay ना? Well, so till here we have completed with the male gametogenesis, microsporogenesis, and in totality we have completed the male reproductive system of flowering plants. Any difficulty in understanding any point, beta? Any point not understood? All of you all have understood everything. That's good. So the next, as you all know, that as we had done in human reproduction, we had first started with what? The male reproductive system. And then we had progressed ahead with understanding the female reproductive system. In the same way, even for this chapter, in plants also, we have completed the male reproductive system. And we have to move ahead with understanding the female reproductive system. But to make you all understand the female reproductive system, I require a continuity. I can't just start and stop abruptly because the time would be up within the next 15 minutes. So I'm not going ahead with the female reproductive system right now. I'll continue with that. So even if it was a Friday collection, I would have continued, but it will be a gap of 1 week because we're going to meet directly on next Wednesday. And by the time the next Wednesday comes, हमारे सर के ऊपर से volatile होके चला जाएगा वो, है ना? So I'm not going to start today. I'll start with the female reproductive system in the next class. So for today, whatever I have taught, I think that's enough because it was a completely conceptual content that I need to, like that I have taught you all. And I want all of you all to not mix the female and the male structures anywhere. Okay. So I stop for today and let's meet on next Wednesday with the female reproductive structure of the flowering plants. Is it okay? Chalo. Bye beta, see you all. Enjoy the weekend. You don't have a test on Friday is what I came to know. So you can enjoy this weekend. Okay? Chalo. Bye bye, see you all. Take care. Yes, same to you, my dear. Same to you. Bye bye.